to the 8th talk in the Mathematical and Computational Ophthalmology seminar series. My name is Paul Roberts, and I'm a lecturer in the Department of Optometry and Visual Sciences at City St George's, University of London, where I lead what I believe is the world's first group dedicated to both mathematical and computational ophthalmology. I'm the creator and host of this seminar series together with David Crabb, who heads the Crabb Lab at City St George's, University of London, and Phil Luthert, an Emeritus Professor of Pathology at the UCL Institute of Ophthalmology. This seminar series aims to showcase research in mathematical and computational ophthalmology to the broader ophthalmological, vision science, medical, mathematical and computational communities, raising awareness of the exciting research being conducted in the field and encouraging future work in this area. Do subscribe to this YouTube channel if you'd like to keep up to date on past and future talks. These are also listed on our worldwide page, the link to which can be found below in the About section of this channel. And our thanks go to Worldwide for instigating this platform for sharing scientific research. Before introducing our speaker, Christine Kershier, I'd like to share two items with you. First, the Investigative Ophthalmology and Visual Sciences special issue on mathematical and computational ophthalmology, guest edited by David Crabb, Phil Luthert and me, is now live. 25 papers have been published thus far, including an invited review from Christine, with more to follow. Second, I'd like to tell you about my new paper, which offers advice to early to mid-career researchers across the biomedical, life, physical and mathematical sciences. For this paper, I've compiled advice from 10 past and current presidents of the Society for Mathematical Biology across a range of topics, including deciding if a career in academia is right for you, finding and working with a mentor, formulating a research question, networking, and applying for faculty positions. I hope that this article will prove a valuable resource to early and mid-career researchers as we make the first steps in our academic journey, providing a helping hand from those who have trodden the road before us. Links to both the special issue and the advice paper can be found below in the About section of this YouTube channel. I coined the term mathematical ophthalmology to denote the field which uses mathematical tools to preserve vision by elucidating the mechanisms underpinning ophthalmic health and disease, while computational ophthalmology is an existing term used to denote the field which uses computational algorithms, in particular AI, to preserve vision by identifying and predicting ocular features and risks. Thus, the focus of mathematical ophthalmology is upon elucidating mechanisms, while that of computational ophthalmology is upon prediction and identification. In our last seminar, Giovanna Guidoboni gave a fantastic talk on mathematical and computational modelling of ocular hemodynamics from theory to applications, the recording for which can be found below on our YouTube playlist, together with recordings of all previous talks. This week, we're joined by our speaker, Christine Kershier. If you'd like to ask Christine a question, then please do send these in via the YouTube chat, both during and after the talk. The talk will last for about 45 to 50 minutes, followed by 10 to 15 minutes for Q&A, in which I will relay your questions to Christine. So it gives me great pleasure to introduce Christine Kershier. Christine is a world-renowned vision researcher who has made seminal contributions to the anatomic and molecular pathobiology of age-related macular degeneration, or AMD, which degrades central vision in aged adults worldwide. Using tools of digital histology, her lab has made important discoveries about the composition and role of drusen deposits in human AMD, while also identifying hallmarks such as early loss of rod photoreceptors, gliosis, and RPE transdifferentiation. Her microscopy studies support multiple clinical diagnostic techniques, including optical coherence tomography, autofluorescence, adaptive optics, and rod-mediated dark adaptation. Christine's research has been supported by multiple NIH grants and foundations. She reviews for government and private funding agencies in the US, Europe, UK, and Asia. She serves on the editorial boards of the journals Investigative Ophthalmology and Visual Science and Retina, and has amassed over 265 PubMed entries, over 34,000 citations, and has an H-index of 91. In addition, she has received numerous prizes, including the Ludwig von Salman Prize and Research to Prevent Blindness David F. Weeks Award. Together with Cynthia Owsley, she will receive the 2025 Proctor Medal from the Association of Research in Vision and Ophthalmology. So I'll hand over now to Christine to present her talk entitled Why Age-Related Macular Degeneration is a Mathematically Tractable Disease. Christine.
Thank you very much for the introduction, Paul, and the invitation to speak to your audience today. Please confirm that you can see my slideshow and not the presenter view.
Yes, it's all good.
Okay, good. So uh, I'm happy to be here today. Thank you all for coming to listen. I'm going to talk about AMD as a mathematically tractable disease. I'd like to acknowledge my support over the years, particularly the National Eye Institute, and below are my disclosures for conflict of interest. So here are my takeaway messages that are summarized in this review article that appeared in Paul and David and Phil's special issue, and that AMD is a mathematically tractable disease, especially at its beginning, not at the late stage. And that's because of the neural geometry of the foveal singularity, which provides high dynamic range and huge biologic effect sizes, and in combination with cellular level clinical imaging. We have enough information now from clinical lifespan and evolutionary timelines, so we have another dimension. We have snapshots from tissues, histology, gene expression, analytic chemistry. There's a viable model of how drusen form, and there's molecular commonality with atherosclerotic cardiovascular disease, a field of medicine that has a long history of success. So let's go along this road. Here is the largest the largest risk factor for AMD is by far and away aging. As learned from the population-based studies using color fundus photographs like these, showing the prevalence of all forms of the disease going up after age, especially after 65. Now, the early form of the disease has yellow deposits behind the retina called drusen, as you can see in the fundus photograph, and it can go to either a late atrophic or an exudative end stage. And the way to think about this is an underlying degenerative process and a neovascular complication. Now, AMD prevalence is about twice that of Alzheimer's disease, making it the largest CNS neurodegeneration on the planet. And yet, I'm going to hope to convince you that it's one that's the most approachable. Now, the neovascular form has been treatable with injections of anti-VEGF medicines going back to 2006 now. And just at least in the US, the atrophic form now also has approved treatments for anti-complement proteins. So where I want to go with the presentation today is to consider prevention. Can we maximize the vision health of 200 million older persons? So this is a mathematics seminar. So we're going to talk about X and Y. The three-dimensional Cartesian coordinate system and the layout of the retina is X and Y. Together, the photoreceptors and the RPE form a sensing chip for the brain, like what's inside your smartphone. And here is the chip, and we can take it out and look at it. And we have this in the eye. Here are the cone photoreceptors in the fovea, and here is the RPE as both visible in living people. So these are invisible in people, and they were sculpted over millennia by evolution. These are wonderful chapters about how the rods and the cones in their physiology and distribution match the visual demands of the natural environment for each species, including humans. So we're going to view the photoreceptors in a flat mount at the plane indicated by the line over there through the ellipsoid, all the mitochondria, and viewed as a flat mount. In the image plane of the retina, we can see the photoreceptors of fovea on the top and the perifovea on the bottom, all at the same magnification. And you'd see that the fovea on the top has thin cone inner segments, and the perifovea has 3 times larger cones surrounded by rings of rods. Now, if we look at the same magnification at a cut through the retinal pigment epithelium, we can see that it has geometrically precise cytoskeleton in columns in panels C and D in both places, as well as autofluorescent lipofuscin and melanolipofuscin. These are two long-lasting residual bodies from the lysosome system. So my microscopy of human photoreceptors, shown here, is the basis of single-cell imaging in the retina. Pictures like this provided visualization targets to help launch adaptive optics in imaging like this, which is very amazing. This work also provided complete digital two-dimensional maps of rod and cone density, and this is what turned out to be really important for AMD research. So here are maps of the rods on the left, the cones on the right, and here you can see the important features, the rod ring of high density around the eccentricity of the optic nerve head or the black spot. And then on the right, the high concentration of foveal cones. Now, the 6 millimeter diameter central area, which is an area defined by the high density of ganglion cells, this is the area that's affected by AMD, and it's less than 3% of the total area of the retina. And it's captured in this clinical ruler called the ETDRS grid. We're going to look at a slice through that grid in the next slide. Another important point to make is because of the very small area of the retina that's affected by this disease, all of our assays have to be designed with that in mind. I'm going to come back to that point. So the graph here shows the density of cones, rods, and RPE in color-coded subfields of the grid. The cones here have that high peak, and they have a five-fold gradient in the central subfield, whereas the rods go from 0 to 130,000 per square millimeter just outside the ETDRS grid. Now, in the bottom is a cross-section of the human fovea that's scaled to the grid with the vascular beds shown by the red dots. And the macular yellow pigment is the macular xanthophyll carotenoid pigment, lutein and zeaxanthin that's highly concentrated in the foveal center and in the plexiform layers. In the central subfield and the inner ring of the ETDRS grid are together 3 millimeters in diameter, and they coincide with the macula lutea, the yellow spot. And I think it's if we use that name, the macula lutea, to mean that yellow spot, it'll help us concentrate on important mechanisms underlying the vulnerability of that area for AMD. So the consequences of this highly localized map of the photoreceptors is that the proximity to the foveal center dictates photoreceptor content. And we're going to look at the rod-cone ratio here. The 1 millimeter diameter fovea, which is practically all cones, it has a few rods around the rim. You can see that over on the right on the ETDRS grid. Here's the 3 millimeter diameter macula lutea. It's rod dominant at about 4 to 1. So remember that the macula lutea that's involved in AMD is actually rod dominant. The rod ring of highest density is actually outside the ETDRS. You can see that in the map below. The little arrowhead is showing where the rod densities are highest, and they're near the vascular arcades. It's highest superior to the fovea, where it's about 20 to 1 rods to cone. And I'm going to point out in the next few slides the annulus of vulnerability of aging. You can see that in the ETDRS grid at the top, and that is on the inner slope of the rod ring. In other words, not where the rod density is highest, but where you're coming up the wall of the crater. And that's a disproportionate area of rod loss, where it drops from about 4 to 1 in a young adult to 3 to 1 in an older adult. And these are the data that were used to make that conclusion now over 30 years ago. These are whole mounts showing a young adult and an older adult. And there's a 30% loss of rod photoreceptors between those two images. There are no gaps or holes. The mosaic has filled in the gaps. And this is from a few years later, in normal and AMD eye. You can see in the normal aged eye, a fovea looks like what I just showed you. And in the AMD eye, it doesn't look that different. So the cones are protected in this disease, whereas in the perifovea, you can see the large cones and the intervening rods in the lower left. And in AMD, the rods are depleted and the cones are deformed. And so this is the ring of rod vulnerability in aging in AMD. And what I'm going to show you in the next slide, these two spots are two places where we measure the function of rods in eyes of different ages and different stages of AMD. Testing rod vision near the fovea turns out to be a very cogent way of assessing the state of the support tissues underneath the photoreceptors. This is from landmark work from Lam and Pugh, showing how rod-mediated dark adaptation is rate limited by the availability of 11-cis retinal. That's a retinoid derivative that comes in across from the plaza across all the tissue layers affected by AMD, the RPE and the photoreceptors. And so the test that we use is one of a bright flash of light. And it takes a period of time to recover one's sensitivity after that flash of light. And it's rate limited by the availability of the retinoids. In the study that I share with Cynthia Owsley called All-Star 2, the Alabama study on early age-related macular degeneration, we tested rod-mediated dark adaptation at two different places, and you can see them at the fundus photograph, one close to the fovea and one further away. And if the speed of recovery had been the same in both places, the metric that we use for measuring speed would have lined up along that line of unity of slope. That's rod intercept time. And it doesn't, because rod vision slows more near the fovea than it does further out. So, and that's, remember, that's where the rod cone ratio is rather low, about 4 or 3, as opposed to where the rods are more abundant further out, but it slows more near the fovea. So let's look at the other dimension. We've talked about X and Y, the image plane of the retina. Let's look across the layers. And that's something that's been made possible since the advent of spectral domain OCT, shown here. You can see all these wonderful imaging techniques. Here's the Cartesian coordinate system. There's the XY we've been talking about. Familiar imaging methods like color fundus photography, fluorescein angiography, and OCT gives us the ability to look across the retinal layers. So that's Z dimension. And then even more amazing is this one. It's T for time. And that's because spectral domain OCT is eye-tracked, and it's possible to follow the changes in the disease of a slow-moving disease over time. My clinical collaborators have patient records going back to the inception of this technology. So that's now 16 years of eye-track data. It's a miracle. So let's look at the OCT. You can see this investigational device on the left that has 3 micrometer axial resolution. Now, it's really important to have good axial resolution in looking at the layers, because they're increasingly smaller. The photoreceptors are about 140 microns tall at their longest, whereas the RPE is about 14 micrometers tall, and Brooks membrane that I'm going to show you in a minute is about 2. So advances on AMD are really a function of axial resolution. This is adaptive optics assisted OCT, and you can see all these reflective bands. Most of these have names from the consensus nomenclature, and the hypo-reflective bands between them, only one has a name so far, but we think this will change because we believe that there's an ultrastructural basis to all these because we're dealing with the reflectivity of light, the redirection of light off of liquid water interfaces. So there's an ultrastructural basis. And we put this to work with volume electron microscopy, collaboration with Dennis Dacey and Andreas Polreis. And you can see what the RPE looks like in a total volume reconstruction. And you can see the membrane specializations, the apical processes, the basal enfoldings below, and in between, very precise stacking of organelles, melanosomes, lipofuscin, melanolipofuscin. Those are the two responsible for autofluorescence, and then a dense layer of mitochondria on the bottom. So now I mentioned Brooks membrane. This is the inner wall of the choroid. This is the transport barrier between the choriocapillaris endothelium and the basal surface of the RPE. And it's been historically considered a 5-layered structure, but we think it's best considered as a 3-layered structure in which the RPE basal lamina is considered with the RPE, which makes it, the RPE being an epithelium, has a basal lamina by definition. So there it is, the blue line, and you can see the 3 middle layers of Brooks, inner collagenous, elastic, and outer collagenous. And these are eyes of different ages. In an AMD eye, an older eye, the green now is a deposit, an extracellular material that's largely extracellular matrix. That's called basal laminar deposit, and it and a native basal lamina separates with the RPE, and it opens this space called the sub-RPE basal laminar space, and in there we get drusen, the characteristic extracellular deposits, choroidal neovascularization, it's type 1, and consensus nomenclature, and fluid. And just to emphasize that the genetic influences, the genetic regulation of all these layers are different. So now in the OCT era, we have a 3-dimensional, multi-layer deposit-driven disease. And if we look at that OCT-B scan on the left, and we can recognize that there are 10 biologically distinct layers and less than 100 micrometers of vertical height between the external limiting membrane above and the choriocapillaris endothelium below. Here is the 3-layer Brooks membrane that I just mentioned. And these are the 2 of the clinical end stages of AMD, the 2 neovascular forms, choroidal neovascularization coming from below at the bottom of the schematic, and up above, retinal neovascularization coming from above. I'm using the consensus type 1, type 3 nomenclature, but basically we're talking about choroidal versus retinal neovascularization. And both of these are preceded by characteristic deposits. Down below are drusen, the yellow spots we saw way back at the beginning. They're beneath the RPE basal lamina, between it and the intercollagenous layer of Brooks. And above it, the RPE is another deposit called subretinal drusenoid deposit, or you'll see it in the literature as reticular pseudodrusen. And given my start in vision research with the photoreceptors, to me, one of the most amazing pieces of the story is that the deposits track the topography of the rods and the cones. SDD follows, at least in part, the topography of the rods, and soft drusen material is highly focused under the foveal cones. So now we've defined the regions of AMD, the layers of AMD. That's the X, Y, Z. And now we're going to look at aging, which is lifespan changes. So in the OCT era, we want to see what the different layers are doing. People argue about where does it all start, the RPE, the choroid capillaris, somewhere else. So what we're going to do in the next slide is compare slopes across layers. And what I've done is reanalyze tissue level studies. I've simply taken 20 years old, 90 years old in a group of studies, taken those 2 points and connected them with a line. And we're going to look at the slopes of those lines. And on the left, you can see these are our data showing parafoveal rod loss about 35%. And the cones declined about 8% over that time. This is normal aging. Now, this is not AMD. And you can see that the RPE is actually steady. So the RPE is quite resilient. If we look at the choroid capillaris and the choroid, you can see that the choroid capillaris, it's a planar set of blood vessels, capillaries that cover Brooks membrane, and that goes down about the same amount. In fact, those two things decline together: photoreceptor rod number and the choroid capillaris coverage of Brooks membrane. And in between, if we look at Brooks membrane, we can see that the thickness doubles. The three-layer Brooks doubles in thickness. And this teal line is a esterified cholesterol. It goes up 15-fold. So this is a huge, huge age change. And this is this is one of the main components of high-risk drusen. And it signifies that lipoproteins are present because esterified cholesterol is released from cells only in lipoproteins. I'm going to come back to this. So we now have enough of the XYZ to assemble what I'm going to call the center surround model of deposit-driven AMD progression. And this is the center surround model comes from the mathematics of receptive field organization, which is that you can have opposing mechanisms that are spatially concentric, and they are assembled mathematically in this way. Here's a Gaussian curve. There's a narrow Gaussian, positive going, and you put it together with a wide Gaussian that's broad, and together you get the sum, the bold line there. And as I mentioned, this was how receptive fields began to be modeled decades ago. But it applies here because if we look at this map, which I showed at the beginning, there's the ETDRS grid. And we have protected cones in the center. That's a narrow positive going function. Whereas we have a broader negative going function, which is harm due to the accumulation of drusen and deposits under the fovea over a wider area. And when you put those two things together, you get a center of protected cones versus a surround of vulnerable rods. And the rod-mediated dark adaptation test point that I showed earlier at 5 degrees is right here. So we're probing right at that little area where the rods first come in, but what the rods are doing is telling us what's going on in the fovea under the cones. So I'm going to talk about the deposits, drusen and subretinal drusen deposit, because there's debate as to what the significance of these things are. Are they an epiphenomenon or a precursor, an indicator, or what? So I started out reading the Wisconsin, the Beaver Dam Eye study led by Ron Klein in the 80s and 90s. The baseline for Beaver Dam reported the frequency of early AMD lesions, so that was drusen and RPE changes, reported them by age in the Beaver Dam Eye study. That was 4,500 people who had fundus photos taken ages 43 to 75 years. And they quantified the pathology in terms of that ETDRS grid that I introduced earlier. And you can see the age-adjusted frequency of pathology in those zones. And if they express them as in terms of the area of the subfields, because the central subfield is tiny and the outer ring is big, you can see this quite a bit of more vulnerability in the central subfield. And if you look at the ratio of the central subfield to the outer ring, it's 26-1. This is huge. It's a huge difference. That the central subfield is 26 times more vulnerable than the outer ring. Now, the authors of this study didn't mention that. They reported it, but they didn't discuss it about what it meant. And they didn't really go back to it. But another study designed to be like the Beaver Dam was the Blue Mountain Eye study in Australia. And their 10-year follow-up, they reported the risk of advanced AMD, late AMD, either neovascularization or atrophy based on the location of pathology at baseline. And you can see here that drusen within 500 micrometers of the foveal center, that's right there in the ETDRS central subfield. Just a minute, I did something weird here. Okay. The central subfield, again, it's 26. It's a huge risk right in the central subfield. This is larger than all other population level risk factors. In fact, the iRisk Consortium in Europe determined that all the risk variants of all the genes plus lifestyle together would get you to 26. And what I'm showing in the table above is strictly aging. So this is an enormous biologic effect. And just for reference, smoking, which was determined to be a risk factor for AMD, the odds ratio are about 3. So the biology here, there are huge biological effects focused under the fovea. Now, this analysis separates the central soft drusen from all the other drusen. This is a flat mount. Phil Luther, it's a co-author here. You can see a human retina, all the zillions of drusen out in the periphery, but it's possible to have a clean central area. So those drusen do not confer risk, and that was confirmed last year by analysis of a large data set by Amitha Daltmapalli and her group. And finally, here is the drop-off in the inner ring. The inner ring has risk also, but it's about a 3-fold less risk than that central subfield. So all the risk for progression for AMD is concentrated in the central 3-millimeter diameter. That's the macula lutea. So the fundamental histopathology and ultrastructure of AMD was done by Shirley and John Sarks and Murray Killingsworth, who was the collaborating electron microscopist. And they had histology of hundreds of clinically followed eyes. Dozens of them had transmission EM, like what I'm showing here. And they called what they found the drusen membranous debris because their preparation techniques, that's what it looked like. And they said that this principal component was found only in the central area, but they didn't present data. We presented data in a small number of eyes. We microsected the drusen from the central and peripheral areas, 415 drusen. And you can see over there on the left what the results were, that the central area of AMD eyes had about 3/4 of the drusen found were so-called soft, the high-risk drusen, whereas there were none found in the peripheral area. So this is the confirmation of, although it proceeded in time, actually, a lot of these findings came out in sort of reverse order, but it shows that the central area is much more vulnerable to the periphery. Now, this is not something that's widely appreciated, and I want to point out at least two reasons why. Some very important studies done almost a quarter century ago now. This is Greg Hageman's study showing that drusen, this was really seminal in introducing the idea that complement was involved in drusen formation, but these were mostly extramacular drusen. So the sampling scheme did not include the 3 millimeter central area that was vulnerable. Likewise, Crabb and Hollyfield, the drusen proteome study from 2002, another seminal study, they were interested in markers of oxidative stress. They combined drusen from the center and periphery. So remember that 3% area, unless you sample specifically to get that 3% area, it's very difficult to find. So there are actually many factors in AMD research that can be considered in the light of these topographic considerations. I'll discuss them in the question and answer if anybody's interested. So let's just look at these soft drusen. You can see a big one there, and it is continuous with a layer of the same material. And this is called basal linear deposit by the Sarks, but it's really the same material, and same high risk material. And we named a precursor called pre-basal linear deposit. You can see it in the different color arrows. And I'm going to propose to you, based on some of the things I'm going to show you next, that in order to produce these high-risk drusen, you actually need functional RPE. And when they're present, the drusen form a transport barrier. It puts the RPE into metabolic insufficiency as it pushes the RPE away from the choriocapillaris. Because these drusen are very rich in lipids, it's a site of pro-inflammatory lipids. There's a cleavage plane for type 1 neovascularization and RP hypoxia. And if you look at our website of the AMD pathology, where we had many AMD eyes and controls, you can see up above that the fovea on the left is where a soft drusen material accumulates, whereas the in the perifovea, it's much reduced. And in aged eyes, the precursor pre-basal linear deposit is more common in the fovea than in the perifovea. So based on the aging studies and these topographic considerations, drusen appearance in AMD culminates a process that actually starts in late adolescence, and it's ongoing throughout adulthood under the fovea. So some people ask whether deposits have anything to do with the advanced stages. So I'm going to show type 1 neovascularization, in which drusen are a direct precursor. The Sarks showed this, and here's a cartoon showing the vessels growing in where the drusen are located. Here are drusen and basal linear deposit, and you can see it here. There's a little artifactual separation of the word druse, but you can see the continuity. And down the row, here's the neovascularization. The arrows show a break in Brooks. The asterisk shows neovascularization breaking through with pericytes and leaking fibrin. And in between, in all the same plane, that sub-RPE basolaminar space, there's the fluid. Okay. Now, this is the part that was missing for many years, was how drusen lead to atrophy. And I'm going to show you just the OCT evidence, but now we have multiple examples of histology showing this. Here's a very large druse complex, and this is what it looks like in OCT. And you can see that the RPE, that bright band up at the top, starts out as pretty evenly, it looks pretty even. And over time, you see this ruffling up of that bright layer. You can see clumps of reflective material leaving at the arrow. Those are called hyperreflective foci. You see light shining down through the layer, the denuded RPE layer, and eventually the drusen collapses into atrophy. And you can quantify that in these B scans. You can see a fast rise, a slow rise, and a fast decrease. And the way we interpret this, based on what I'm going to tell you next, is that the drusenoid PED like this grow because the RPE is actually producing a material that normally exits to the circulation, but it's impaired from transit by H. Brooks and choriocapillaris. And these large drusen collapse because production by the RPE is discontinued and clearing processes catch up. So in the era of color fundus photography, then atrophy begins because the drusen collapse. But in the OCT era, where we can follow across layers, the drusen collapse because the atrophy begins, because the RPE is dying. And so what I take away from this is that the RPE is actually relatively healthy and doing its jobs, and the transport across Brooks membrane and the choriocapillaris is impaired. And recently, it's been shown that the height of the drusen is strongly related to the features of atrophy. So the RPE on the top of those drusen are far away from the choriocapillaris, and they're vulnerable. So I'm kind of getting low on time here. So what should let me talk about how drusen form, and maybe I'll stop there and I won't get to everything. And the hypothesis about how drusen form is that these are unneeded lipids from a lifetime of feeding foveal cones that are constitutively repackaged as large lipoproteins and blocked as transit to circulation. And the way to think about this, at least I do, is that two dials control the deposit formation. Here's a sink with the outer retina dripping out into a drain, which is the choroid going out to the systemic circulation. And here is there's a large inflow to the RPE, meaning giving a large outflow out to the circulation. And the large inflow is the outer segment phagocytosis, which we've known about for years. But also, I will submit to you that it's the delivery of dietary essentials. So there's a constitutive release of material from the photoreceptors, and there's flow control of the output to the circulation. And just to emphasize the dietary essentials, the photoreceptors have a prodigious appetite. Every day, they shed a piece of their tips, the outer segments, and every day they have to make new membranes. Where do all those new membranes come from? They come from lipids that are delivered from the diet and then processed by the RPE and sent over. So there's a huge dietary need for lipids. Now, as I mentioned way back in the aging graph about esterified cholesterol, these data come from Philippine histochemistry, a beautiful marker for forms of cholesterol. And you can see esterified, unesterified cholesterol in all the membranes of the cells of the retina, whereas esterified on the right is exquisitely localized to Brooks membrane. And the only way you can get that is through lipoproteins. And this is what these lipoproteins look like in electron microscopy of tissue that's preserved in a way that shows extracellular lipid well. And on the left, the pre-basal linear deposit, you can see in the lower left-hand corner all these spherical particles with surface and core morphology of lipoproteins. And the RPE expresses lipoprotein hallmark genes. It's in the business of making lipoproteins. And you can see it there at the arrow. This is our model of what the lipoprotein looks like. And most of you, I'm sure people listening know about LDL. It's the common with the protein in the plasma. It started out as VLDL made by the liver. And our hypothesis is that the RPE makes a VLDL of its own, and that's why there's all this lipid in Brooks' membrane. Recently, a mouse model has been made in which one of the key genes was knocked out, and there's a strong retinal phenotype with an RPE knockout of this gene. We know from tracer studies by Irina Pikaleva's lab that somewhere between 1/4 and 1/2 of the cholesterol coming into the retina comes from diet. And this is in comparison to the brain, where it's only 6%. So the retina has a huge dietary need. And compositional studies, I'm not going to get into this because it's getting late. The fatty acids implicate diet because of the type of fatty acids. So this is one piece of diet that's very important for the fovea, and that's the delivery of the macular xanthophyll pigment, lutein and zeaxanthin. So we know already that there's a strong dietary input to the fovea. And this is a mechanism that we proposed where the RPE is taking up plasma lipoproteins to strip out lutein, zeaxanthin, and a lot of other things and ship it over to the retina. And then it has a packaging problem. It has to get rid of all the lipoproteins remainders. And that's what is repackaged, sent out by the RPE and piles up to create high-risk drusen and increased risk for neovascularization. So I'll finish with a few. I'll just stop because I have too much stuff, as always. Outer segments are actually not required for drusen components. It's something that people thought about for years, but they're not required. And Link Johnson first showed this years ago, that ApoE and a prominent drusen component can be released by cultured RPE, well-differentiated cultured RPE. And you can see that right there. And it looks very much like, on the right, what we saw by electron microscopy in Brooks' membrane. Our group at UAB, Clyde Gidry was the cell biologist, showed with pig RPE, fresh primary pig RPE, you could get a layer of deposits. No outer segments were required. You could see the lipid stain oil red O, and it's where the cells are releasing lipids down into Brooks' membrane. And there is the arrow right here showing the pores, the material in the pores. So you don't need outer segments or RPE injury. What you need is functional RPE in an appropriate culture medium and a transport barrier. And finally, to finish this line of thought, you don't need the neurosensor retina at all. This is accumulation of a soft drusen under a macular hole. So macular holes are something that unfortunately shows up often in a retina clinic. It can sometimes be repaired surgically, but you can see that in this place that's missing a fovea, the system of taking in lipid and repackaging seems to be ongoing, and this is over 7 years. And I've now encountered other clinicians who've seen these things. We have another case that is equally informative. So I'm going to just state kind of my final. I have a whole section on this. I'm just not going to talk about it. But there's a second layer of deposit called subretinal drusenoid deposit. And it is related to the topography of rods. And in fact, I would say that it's what makes a story about the foveal cones believable, because the rods and the cones evolved for vision. The rods were a late arrival, the cones were there first. And there's a whole other set of deposits and risk factors related to them. But at this point in time, we can connect the evolutionary biology of the retina to the clinical presentation. And there are huge biological effects, huge risk ratios, and that means that there are huge prevention and treatment targets. And that's why I would say that AMD is a mathematically tractable disease. So I'm going to stop here and just take questions. Thank you.
Well, thanks ever so much, Christine, for a fantastic talk. That was wonderful, and I can see that it's already stimulated a number of questions. If you haven't written a question, or even if you have and you'd like to write another, then do please type them in, and I'll relay those to Christine if we have time. So we have some already. So we have. I may not be getting these in quite the order they came in. Apologies if I don't. So one here from John Doe. In fact, he's written three. So the first is, in longitudinal studies using SDOCT to monitor AMD progression, what are the challenges in ensuring consistent imaging quality and accurate registration of serial scans?
Well, these are really good questions. Thank you for asking. There are now several population-based studies using OCT as the image capture device. What I showed you, the older studies using color fundus photography, these are still great resources for a lot of information, but now they're being redone. Particularly, there are a couple of in Germany for OCT. And it's important to get consistent imaging, and it takes experience with imaging, a good centration. It's even more important with other technologies like fundus autofluorescence. Need good focus. So trained personnel and experienced personnel are probably the single best thing that you can use that are important for quality assurance. I remember Ron Klein telling me that he spent 25% of his budget on quality control for Beaver Dam. I see the questions here, Paul. Should I continue, or are you going to read them out?
I'll read them out so that everyone hears which ones are coming. So perhaps I'll do a second one for John, because it relates to this his first question. So the second question was, how do these changes affect the interpretation of changes over time, particularly in detecting subtle disease progression?
Subtle. I've been involved in a lot of the validation of OCT over the last decade, and these changes were not subtle. There were things like crystalline deposits, fluid, outer retinal tubulation. It's a scrolling of the photoreceptors. So there's actually a good deal of validation for advanced stages. With the All-Star study, we're interested in AMD onset and early progression. There, it's a bit more difficult with OCT. We know from a deep learning study that changes of importance are focused in narrow planes on either side of the RPE and the photoreceptors. So these may be end up. We may be going with a metabolic type imaging technology at early disease. It's still very fluid. But progress is being made. I think that there's a kind of a community agreement that atrophy, even if people they're excited about the approvals, that we really need to treat earlier. So I'll stop and go on to the next question.
Thanks very much, Christine. We've got a question from Mukit Kulman, sorry, apologies if I'm mispronouncing this, Kulmaganbetov. He asks, is it possible to get the slides? I think what we said discussing this before is that if people want specific slides, you can email you personally and discuss whether that would be a possibility. Is that okay?
Yes. People certainly can get in touch. And then this is the nice thing about your format, Paul. It'll all be available online. So
Yeah. So yeah. So yeah.
Yeah.
So a recording of this talk will be maintained at the same link on YouTube. So if you want to see this again via that video, then that will still be available. The same person, Mukit, also asks, what do you think about the polarization perception in patients with AMD?
So this is, I assume that questioner is asking about like Heidinger's brushes. It's like an optical illusion that you can see that are set up by the polarization of light in the Henle fibers. Now, the Henle fibers are the angled fibers of the photoreceptors and Mueller glia that connect from the cell bodies to the terminals of the photoreceptors, and then to connect to the rest of the circuitry. So I know that there is some work on polarization perception, but I don't know the current state of that. So I mean, it sounds like something that's worthy of study, but I really don't know the details.
Sure, thank you, Christine. And then there's another question, again from Mukit. He says, macular pigments can be found in several retinal layers. What do you think about the protective properties of MP in ONL against MP in HFL?
I mean, at this point in time, we really don't have a basis for saying one part of the macular pigment distribution is better than the other part. Now, I recently published with Dennis Dacey and Andreas Pollreich a paper quantifying Mueller glia mass in the fovea, and it's very high in the layers that are rich in xanthophyll pigments. A whole line of research that I couldn't get into really established the Mueller glia as an important repository of the xanthophylls. And so the xanthophylls are probably doing something very important on their own, but insofar as that they're a marker for the Mueller glia are providing a lot of support services for cone vision. So this is an area that I think is ripe for new research, especially with the new imaging technology. And I should say things like single cell RNA sequencing. I mean, now we know what genes are expressed by a lot of these cells. So that's exciting.
Thanks, Christine. So going back to John Doe, so he asks, regarding spatial concentric opposing mechanism, can you elaborate on the specific equations or models used to capture the interaction between these opposing forces and their impact on the retina?
I'd love to be more specific, but we don't actually have the numbers yet. I mean, I simply borrowed the difference of Gaussian paradigm that was used for visual fields, because qualitatively that shows the protected cones and the vulnerable rods very well. Now, how that would actually work, I think we have a better chance of determining what kind of numbers would fit into the drusen part of that, because I mean, we published volumes of the lipid layers, the pre-basal linear deposit and everything. So we have numbers for that, or at least static. But we don't have numbers yet to quantify the protective side of that, the positive going part of the curve. So in the review article that I mentioned, we said that this is an area that we hope will stimulate research, because that it could be, our qualitative assessment certainly could be quantified with more information over time.
Thank you. And then John goes on to ask, how does the absence of outer segment involvement influence our understanding of the role of the RPE in drusen formation?
Well, let me just, that's a good question. Let me preface what I'm going to say by that kind of dramatic video of the drusen bubbling along under a macular hole. The RPE may be contributing to drusen, but I doubt that they have the same composition as a regular drusen, because the outer segments, the lipids from the outer segments do eventually end up in drusen. Particularly cholesterol is a likely component, but we don't have the exact circuitry of that yet. We don't have the tracer studies. My model of how drusen form is that the RPE is doing its job, a constitutive job, and it's doing that until as long as it can. And that releasing lipoproteins to circulation is part of its activities. And if you look carefully at most of the drusen formation hypotheses in existence, they're usually something to do with lysosomes, which, I mean, lysosomes are involved, but they don't drive the whole system, or that it's an RP injury response, that the RP is injured in some way and it produces the material in drusen. And I would say that the RPE is healthy, producing this material that's stopped up by the action of Brooks and corticapillary. I mean, from the perspective of deposit formation, the whole thing is most easily explained. If I'd gotten to the end of my talk, I would have shown this. Most easily explained if the initial step is failure of the corticapillaris endothelium.
Thank you. And perhaps this question overlaps slightly then with some of what you said there, but so it may be that you've not got anything to add here, but I'll ask in case you do. So the next question was, are there specific metabolic or secretory pathways within the RPE that are critical for the accumulation of drusen materials?
I mean, I tend to view it in terms of lipid transfer. And in part that's because because of our initial studies were done with histochemical localization of cholesterol and its forms, and electron microscopy, which shows electron-dense fatty acid. So I think that lipid transfer is an important pathway. And some of the proteins, like ApoE, is plausibly part of a lipoprotein particle. But lipoproteins carry more than just apolipoproteins. Plasma HDL has a lot of complement proteins on it. So I think that we're still at the early stages of the intracellular mechanisms that lead to a lipoprotein coming out. I'm very excited about Cassie Bose-Battaglia's knockout mouse, because it means that some of the regulation of these pathways can be studied experimentally.
Thanks, Christine. So another question, again from John, is, does the exclusion of outer segments from the drusen formation suggest alternative pathways or mechanism driving the transition from early to advanced AMD?
Well, early passes through intermediate, so you have to get there. I don't want to leave the impression that outer segments are excluded. Outer segments are not the driver. They're not required, right? That's a difference, okay? I think diet is required, and outer segments are participate, but they're not the driver. Now the transition from early to advanced, those are, I mean, a lot happens in those spaces. I mean, clinically, there's a lot of interest in predicting what eyes are going to advance in geographic atrophy, what eyes, because now there's an approved treatment, at least in the US. To me, the kind of the frontier of where to approach is the beginning of intermediate, is the transition from early to intermediate. Because at least in All-Star 2, we're seeing changes in autofluorescence and corticapillaris flow signal density at that interface. So I think that over time, treatments will be applied to earlier stages of disease, and uh, that's a good thing. More people can benefit.
And then the final question, which perhaps overlaps a little bit again. So, how might this understanding affect current or future therapeutic approaches targeting drusen or AMD? For example, should therapies focus more on the modulation of RPE functions or ECM composition rather than photoreceptors?
Well, it's a big disease, and there are a lot of potential targets. I mean, when we were very active in researching about lipoproteins, we proposed the oil spill strategies, and the oil spill to clean up the oil spill, and you can do that with lipid scavengers. To scavenge oxidized lipids. You can modulate what comes out of the RPE. Statins or NASH inhibitors, there are several ways to do that, and then to modulate through the diet. So that's just the lipoprotein pathway. There are other approaches to either restoring corticapillary health, maintaining health, reducing the adhesion of lipoproteins on Brooks membrane. Why do they stick there? Why don't they just go on out? So, I mean, there are lots of opportunities, and even I'm not sure what the questioner is thinking about in terms of photoreceptors, perhaps a neuroprotective mechanism. And well, I mean, a neuroprotective mechanism might have some merit if in conjunction with approaches to targeting the tissues that are more directly involved in the disease and the problems with transport, which I think are the main ones.
Thank you. And then there's a question here. I think we'll make this the last question because we're going a little bit over time. So this is from Robert Cooper. He asks, so it was noted that choriocapillaris thickness decreases along with rods. It was also noted that the Brooks membrane function was impaired. What are your thoughts on the relationship between the two? I.e., do you think that the choriocapillaris is shrinking because of impaired Brooks membranes?
I think that I mean that's a good question, because I have a feeling that we're going to be able to get more and more specific about these changes in Brooks and choriocapillaris endothelium. But at this point, I think we don't have enough data. I mean, when you look at the quantitative studies of choriocapillaris transport, there are very few. There are very few. Phil Luther, I don't know if he's watching today, but he's involved in some very nice quantitative studies with Musa Zawash, Brad Gelfand. So we really need to know more about how the choriocapillaris works, because it may be, I mean, the choriocapillaris endothelium, these are cells, they have receptors that are potentially druggable, or it may be more conceptually easier to consider intervention at that level. But I mean, really, this is speculation on my part, because we really do need a whole lot more data. So, and I hope people on this who are listening will go and get it. That would be great.
Well, thanks. As I said, we'll make that the last question. Thanks ever so much, Christine, both for a really fabulous talk and for all your time in answering each of those points. I'll just close now by saying that further details of all future talks can be found on our worldwide page, the link to which can be found in the About section of this YouTube channel. And do also subscribe to this YouTube channel if you'd like to keep up to date on past and future talks. So thank you again to Christine. Thanks to you for listening, and see you next time on mathematical and computational ophthalmology. Goodbye.